Where does replication begin? There must be some place in the DNA where this process starts. Surely it's not just random. Replication origins are actually recognized. The sequence is detected and there's a specific component of that replication machinery that has this recognition function called DNA A, which is shown here. The other components, for example, helicase and single-strand DNA binding proteins initially, are then recruited to this site where replication initiates. There's only one site in prokaryotes. In contrast, there are multiple sites in eukaryotes. Why might that be true? Think about differences in magnitude of the replication process for these types of organisms. Methylation in eukaryotes, which is a reflection of other things going on in the cell, is an important part of timing a replication in higher organisms. DNA A is the protein shown here bound to DNA. It recognizes sites that are the points at which the DNA is first separated and the first RNA primer is made. DNA A recognizes the origin of replication and forms a complex with helicase to open the strands and allow DNA primase to come in and begin the replication process. They are able to open the DNA at the origins because sequences at those sites are often AT enriched, and that presumably lowers the energy needed to make the opening. In prokaryotes, the single origin is recognized by DNA A, helicase, and the DNA G primase. Eukaryotic primases, within a larger complex of proteins and at multiple origins of replication, are not as specific as those in prokaryotes. We looked at where replication starts, but when does this happen? In prokaryotes, which are living free in the environment, they can replicate their DNA when it is environmentally conducive to do so. And this is generally impacted by access to nutrients, to food. On the other hand, in eukaryotes, there are cell cycle signals that determine when replication occurs, and it occurs specifically during the S phase, named S phase for synthesis because DNA is synthesized at this time. The timing of replication in eukaryotes is also affected by packaging of the chromosomes. Remember that areas of chromosomes are packed differently, and that there are very tightly packed regions of DNA and regions of DNA that are looser. The more accessible regions are replicated first, and the more dense regions of chromatin are replicated last. In eukaryotic replication, you've got lots to copy, and you want to do it in an efficient manner. So multiple sites start at different times. And we can look at that here along chromosome 2. You can see the origins of replication initially marked in red, and then synthesis is complete. And we'll look at that again. Different sections start at different times, and some sections move faster than others. And that has to do with dealing with the structure of the chromosomal material. If it is tightly packed, it is going to go somewhat slower. If we look at the human chromosomes and where replication begins, you can begin to get a sense of the differences. This helps you see how replication begins at different places and different times from multiple replication origins. And again, you're supposed to be thinking about why so many? Why do eukaryotes have multiple origins of replication?